So this talk is about overcoming some of the limitations that limit photosynthetic efficiency. And let me start. Well, let me move to the next slide. Another regulatory issue is Zoom. So just to remind you that photosynthetic carbon assimilation is the basis of life on Earth, and everything a plant can do in terms of producing seeds, biomass, defending itself against biotic or abiotic stress, a priori depends on the availability of chemical energy. And this chemical energy comes from photosynthesis, the conversion of light energy into electrochemical gradients, which are then used to generate ATP and NADPH. And this is then longer term stored in the form of triose-phosphates, which are generated from 3-phosphoglyceric acid, which is the first product of the carbon assimilation by the enzyme Bisco. And these triose-phosphates that are produced in the chloroplast or in the cytoplasm in cyanobacteria mostly go back into the regeneration of the carbon acceptor, ribulose 1,5-bisphosphate, but approximately 15% can be taken out. And that's where all the biomass on planet Earth comes from. And that biomass on planet Earth is approximately 450 gigatons of carbon just stored in plant biomass. But it's huge. It's 1/4 of the total carbon that is in the atmosphere. And this is plant biomass. This is a climate one level photosynthesis here. This is the amount of carbon that is emitted from fossil reserves every year. Approximately 10 gigatons of carbon are released to the atmosphere every year. Roughly 3.3 gigatons are drawn down into vegetation, into permafrost soils and things like that. 2.8 gigatons go into the oceans. And this leaves approximately plus 5 gigatons of carbon that go into the atmosphere every year, still now. Okay, this is where photosynthesis actually is. It's the fine print in this representation. 130 gigatons per year are assimilated by photosynthesis, but these 130 gigatons are more or less released again through respiratory activity. So this is more or less balanced, right? And if you can make this drawdown of CO2 from the atmosphere a bit more efficient, so that some of it can be removed and stored, rather than be respired again and CO2 be released again, then I think we can change the 3.3 to a higher value and potentially decarbonize, well, get to net zero and eventually decarbonize the atmosphere. But it also shows the scale of photosynthesis. The atmosphere contains right now 900 gigatons of carbon, and 130 gigatons of this carbon pool are every year drawn from the atmosphere into the biosphere. Then it's released again. So it means every 6 years, the entire carbon pool of the atmosphere is recycling once through the biosphere. This is the scale of photosynthesis. But this is the only process that operates at a scale that is large enough to do something against climate change, in addition to avoiding emissions, of course. There's, however, room for improvement in photosynthesis because only 1% of the light energy that is available throughout the growing season is actually converted to biomass. 99% of the energy that a canopy captures or can capture is not stored in biomass. This is from reviews from Blankenship and Xinguang Su a few years ago. Theoretical limits are around 4.6% for C3 plants and 6% for C4 plants. So there's already like a large gap between what is observed in the field, and the truth is in the field, as we heard in the previous talks, and what is theoretically possible. And so part of my lab works on closing this natural gap. And I won't go much into this, but I think we can beyond these theoretical limits that are existing in naturally existing plants. So what are the targets for improved efficiency? The light harvesting, of course, and that goes from the initial reactions in the reaction centers to the canopy architecture, far red absorption, which is actually quite cool, so that it's possible to absorb far red light and use it for photosynthesis by extending the spectrum of the chlorophylls into the chlorophyll, but also like maybe closing the green gap and harvesting more light, right? This would make black plants, which would be really cool because it looks just cool, but potentially less, a bit controversial with the general public. Pigmentation, leaf angles, this was pointed out, I think, by Anja and also by Loic in the beginning. Like the way how the plants orient their leaves depends, affects the light harvesting. The leaf anatomy, morphology, like things like mesophyll conductance are still under researched and not sufficiently understood so that we could actually screen for it at the genetic level, right? And look for germplasm that has lower mesophyll conductance. Stomatal conductance, the dynamics of stomatic closure, the leaf complexity, like how many layers of mesophyll we have, and so on. Then on the carbon fixation side, it's the Kelvin-Benson cycle. Christian Rains has done a lot of work on this, and she showed that cedoheptulose bisphosphatase is one of those enzymes that release the Kelvin-Benson cycle. There is C3, C4 intermediate photosynthesis and C4 photosynthesis, photorespiration, and there's also new to nature pathways. And in my talk, I will focus mostly on photorespiration and new to nature pathways and only mention in passing the C3 C4 stuff. So for anything related to carbon assimilation and efficiency of carbon assimilation, it's important to understand the process of photorespiration. Many of you will be quite familiar with that, but some are maybe not. So that's why I have a few slides on that. So the enzyme that fixes carbon, ribulose 1,5-bisphosphatase, can also fix oxygen. And at ambient temperature in a C3 plant, it happens approximately 25% of the time. And this number can go up dramatically when, for example, stomates close. The CI, the leaf internal CO2 concentration drops. And this can reach like almost 100% of the Rubisco reactions then become oxygenation reactions. And this oxygenation reaction generates, in addition to phosphoglycerate, also 2-phosphoglycolate. And this 2-phosphoglycolate is an inhibitor of trisphosphate isomerase and cedoheptulose bisphosphatase. And if not removed immediately, it will actually block the action of the Kelvin-Benson cycle. And removing that requires a pathway that runs through several cellular compartments. The starting product is phosphoglycolate. It's dephosphorylated inside of the chloroplast, and then the glycolate leaves the chloroplast, goes into the peroxisome, is converted to glyoxylate. You can read it. Glycine, and then glycine goes into the mitochondria, where 2 molecules of glycine are converted to serine, and this releases CO2 and ammonia. Serine goes back via the peroxisomes, hydroxypyruvate, glycerate, and then glycerate can enter the chloroplast again, is activated to glycerate through phosphate, and then can go back into the Kelvin-Benson cycle. I should mention, because I'm blamed of showing the photorespiration as a closed cycle, and indeed it is not. It has been shown by several labs that it's not a closed cycle. Actually, in particular, glycine and serine are taken out of this pathway. Photorespiratory pathway in the C3 plant is actually the main pathway for glycine biosynthesis in the leaf, for serine biosynthesis in the leaf, and also for C1 units, as we learned in an earlier talk. So the reaction that generates most of the inefficiency in photorespiration is the conversion of 2 molecules of glycine to serine. If 2 molecules of glycines, 4 carbon atoms are converted to serine, 1 is lost as CO2, and this CO2 needs to be reassimilated, costs 2 ATP and 3 NADPHs to bring it back into the reduced form, and it releases ammonia, which also needs to be reassimilated, requires ATP and reduces pyridoxine. So a lot of extra energy is required to deal with that loss here. And if you calculate this for the 25% photorespiration that happen in the C3 plant pretty much all the time in the field, this almost doubles the cost for making sucrose. So making one molecule of sucrose requires 67 ATPs and 42 NADPHs in the presence of 25% for the respiration, and only 37 ATP and 24 NADPH without for the respiration. So there's a huge gap in efficiency here. It has not always been like that. In previous times, the CO2 concentration in the atmosphere was a lot higher until approximately 100 to 50 million years ago. And then around that time, there was a massive decline in the atmospheric CO2 concentration from approximately like 1,000 ppm, which is high enough to suppress for the respiration to very low levels, to the pre-industrial 277 ppm. And most of this drawdown here in CO2 is not due to biological activity, but to geochemical activities. This was the time when the Indian subcontinent crashed into the Eurasian, whatever, like two continents collided, and this pulled up the Himalayas, exposed a lot of fresh rock, and the weathering of this rock led to a massive decline in the CO2 concentration of the air. And that's approximately the time when the C4 plants evolved. That's the only slide on C4, I think. So that C4 is one of those natural adaptations to low CO2 in the atmosphere. C4 is a biochemical carbonate pump or a CO2 pump that increases the concentration of CO2 at the site of Robisco to approximately 600 microliters per 600 ppm. And that is high enough to suppress for the respiration to like 1 to 5% of what you see in the C3 plant. C4 is complicated. Many labs have tried to engineer it, for example, into rice or into wheat. So far, it has not succeeded, but it's still ongoing. A large part of my lab is working on the early evolutionary stages that lead from C3 to C4 via intermediary stages. And we think that this understanding of evolution, of the first steps going from C3 to C4 intermediates, is really important if you eventually want to engineer C4 or even C4 intermediates, which are evolutionary stable intermediates in their own right. And we do that via genomic comparisons, engineering, and things like that. But I'm not going to talk about this anyway. So there are some constraints on evolutionary adaptation and acclimation. If we look at this genotypic variation against fitness map here, selection always goes towards eventually to an optimum. And once a certain optimum has been reached, and there would be another optimum, like a more global optimum here, and in between A and B would be a fitness valley, it would be very difficult to reach this global optimum from the local optimum via selective processes, be it breeding, be it natural selection. So it means that some better parts are not reachable. And this may have to do also with genetic Pareto optimality fronts. Like, for example, if you look at two traits, this, for example, may be the CO2 assimilation rate. Trait 2 might be the water loss from the leaf. If we get maximum CO2 assimilation, we would also get the maximal water loss. Okay, the maximum water use efficiency you would get at no CO2 assimilation, which is also not ideal, right? And from this we get a Pareto optimality front, and we can maximally reach this front, and this point here would be inaccessible because of epistasis, because of trait interactions, and so on. If we really want to go beyond the Pareto front, we need to do revolutionary innovations. We need to go beyond what's present in nature. And Aaron Bar-Evan, the late Aaron Bar-Evan, has eloquently phrased this a few years ago as, instead of asking why the existing metabolic pathways evolved the way they did, our goal is to take advantage of the repertoire of known enzymes to design better pathways for human needs. And I think this quote can now be extended to includes not only the repertoire of known enzymes, but also the repertoire of unknown enzymes that do not exist in nature. And this statement by Aaron is really driving the things that I'm going to talk about. Aaron passed away in 2020, but together we started a Format Plant project and later on the Gain for Crops project. And in the Gain for Crops project, we continue Aaron's legacy. So everything I'm talking about in the next part of the talk is really going back to Aaron's initiatives. Maybe not this because this was before, but anyway. So a lot of this has to do with bypassing photorespiration. And bypassing photorespiration means to not run photorespiration through the peroxisomes and the mitochondria, but do everything in the chloroplast, okay? So go from phosphoglycolate to something else inside of the chloroplast without taking the detour to the other organelles. And the first concept was published by Christoph Peterhensel, who he also delayed Christoph Peterhensel. He passed away last year from muscular atrophy. Is a terrible thing. And they showed that a bacterial glycerate conversion pathway can be introduced into the chloroplast and used to bypass photorespiration. And they observed a biomass increase in the range of 15 to 20% in Arabidopsis in the greenhouse. Veronica Marino, also with some contributions from our lab, showed that a glycolate oxidation cycle in Arabidopsis thaliana chloroplasts also leads to growth improvements under certain conditions in the greenhouse. I should always say as a disclaimer. The big next step forward was when Don Ort's lab, led by Paul Saus, the postdoc in this project, actually took it to the field, because until this paper, all of these bypass concepts had been in greenhouses and growth chambers. And what they did here was to take these different bypass concepts that had been developed by others earlier, the tetronic semialdehyde pathway from Peterhensel, the glycolate oxidation pathway developed by Veronica Marino, and a derivative of this pathway that doesn't use the glycolate oxidase, but the glycolate dehydrogenase, which is called the AP3 pathway. And they put this into tobacco and did field trials with these tobacco plants. And they could show that this is change in biomass in the field, that the largest biomass increase was achieved with the AP3 pathway that goes as a direct glycolate oxidation pathway in the chloroplast. I think these plants have been measured before they set flowers and seeds because of regulatory issues. So it's not entirely clear whether they produce more seeds, but these pathways have now been also introduced into other plants, and they seem to also produce higher yield in the field. This paper here came out in the same year, was published in Molecular Plant. And here the authors included, also put in a glycolate oxidation cycle that completely converts glycolate to CO2 inside of the chloroplast with a different enzyme than in the Marino paper. And they put that into rice, and in the field they could reach up to 27% increase in seed in the spring seeding season. In the fall seeding season, the yields were lower. They have, in the meantime, made a revision of these constructs and included, used now photosynthetic promoters instead of, I think, the constitutive promoters that they used here. And with this revised construct, they see increase in seed yield in both seeding seasons. And that was done at a density of 250,000 plants per hectare and a yield of 5 tons per hectare, which is quite good for rice. So these are really field relevant conditions. So it works in the field. So these current bypasses convert phosphoglycolate to CO2. And the CO2 is then reassimilated inside of the chloroplast. And it's a little bit counterintuitive why it actually works. Because, you know, reassimilating the CO2 requires, 2 molecules requires actually 6 molecules of ATP and 6 molecules, no, whatever, like something like that, a lot of ATP. And there's pathways that should be theoretically better, like the hydroxyaspartate cycle that published a few years ago. This comes from marine bacteria, these marine bacteria can assimilate glyoxylate that potentially comes from cyanobacteria that's secreted and convert it via this beta-hydroxyaspartate cycle to oxaloacetate, which then goes into biomass and energy production into the gluconeogenic pathway. Again, there you would release one CO2 in the gluconeogenic pathway, but you gain actually 3 carbon atoms. The other one is the tacronyl-CoA pathway, or called TACO pathway, in which glycolate is first converted to glucolyl-CoA, and then a glucolyl-CoA carboxylase converts into tetranyl-CoA. This enzyme does not exist in nature, and tetranyl-CoA is further converted to tetranate, and then to glycerate by the tetranate-CoA reductase. Is also an enzyme that doesn't exist in nature, and I will talk a little bit more about these. But before, if we look at the energy budgets for these different photorespiratory shortcuts, you would see that pathway that South et al. Showed to increase biomass in the field under Illinois field conditions is actually not very energy efficient. It's actually much less energy efficient than the wild type situation. What is shown here is the amount of ATP and the amount of reducing equivalents needed to make 1-glyceraldehyde 3-phosphate. And the AP3 pathway is out here. Here is the wild type, here's the beta-hydroxyaspartate cycle, and here are different versions of the TACO pathway. These are different versions of the carboxylase enzyme. This is the theoretical photosynthetic efficiency improvement relative to the wild type of these different pathways. Here would be the beta-hydroxyaspartate in peroxisomes, in chloroplasts. BKs I'm not going to talk about. Here's the AP3, which has a higher cost than natural photorespiration, an optimal TACO pathway and different versions that we already have in plants. So why do these pathways actually work if they're less energy efficient than the wild type? And we address this by computational modeling. This is a collaboration that was led by Marvin van Alst, who was a postdoc in Ed Smith's laboratory, and he's now in Oxford working with Lee Sweetbluff, Oliver Ebenho, and Matthias Heinemann in Groningen. And they looked at the carbon fixing, carbon neutral, partially decarboxylating, and full decarboxylating bypass models in 3 different models: a consumer model, a genome scale model, and a kinetic model. And they looked at the velocity of the carboxylation reaction over the oxygenation reaction, the rate of carbon export, energy limitations, and CO2 limitations. And this is on bioarchive and will come out in Science Advances very soon. So this is the efficiency change relative to the wild type. And up here you see again the TACO pathway, the arabinose 5-phosphates shunt, xylose shunt. These are pathways that I think have not yet been implemented in plants. Here is the Kibish-South AP1, the BHAC and the plastids, the Wang pathway, the decarboxylation pathway. Here's the South AP2, the Meyer and South AP2, and Shen pathway. But you see carboxylation pathways are not very efficient, but they are nevertheless working. So why are they working? When we now look at, here is 100% relative to the wild type. We're looking at carbon use efficiency, carbon export, the ratio of the carboxylation reaction to the oxygenation reaction, VOC, VO, and VC. And what we see is that in these full decarboxylation pathways, the rate of the oxygenation reaction is lower than in the wild type. Like here would be 100%, and for VO, the oxygenation reaction is lower. The carboxylation reaction is higher. And this is also, like, of course, visible then here in the ratio of the 2 reactions. So it means by releasing CO2 inside of the chloroplast, right next to Rubisco, we shift the ratio of oxygen to CO2 inside of the chloroplast, apparently into a direction that suppresses the photorespiratory reaction. So that is apparently the gain of the pathway. So it's really the increase of CO2 concentration inside of the chloroplast, at least based on the model results, that makes these pathways work. Of course, the other carbon fixing and so on are still better, but that's why they work. Marvin and Ed also looked at the carbon export relative to the wild type under different conditions, low stress conditions, ideal like enough light, enough water, and so on. Of course, then all of the bypasses are good. Under CO2 limited conditions, all of the bypasses are outperforming the wild type, but under energy limited conditions, only the less decarboxylating or carbon positive bypasses are better than the wild type. So the initial versions of the bypasses in particular work under carbon limited conditions. That's when the stomates are closed. They don't work well when the sun doesn't shine. It's not a lot of light. And they work particularly well when there is low stress. So I'm still good in time there. Okay, so now I come to the two different versions that we have implemented. The beta hydroxyaspartate cycle that we first implemented in the peroxisomes. And the idea was the entry metabolite of the BHAG is glyoxylate. And glycolate is entering the peroxisomes, is converted to glyoxylate by the naturally occurring peroxisomal glycolate oxidase. We get our glyoxylate. And then together with glutamate, this can be converted into glycine, and this then goes here into the cycle, and out comes eventually oxaloacetate. And we put all the enzymes on one construct under photosynthetic promoters, and we put this into Arabidopsis plants, and we made smaller Arabidopsis plants. So this is the Columbia 0. Wild type, and these are the two selected independent transformants, and they are a lot smaller than the wild type, but they grow as well as the wild type under high CO2, which indicates this is indeed an effect of the photorespiration. Okay, when we transform that into a photorespiratory mutant, and this is here done with the GGT, the peroxisomal aminotransferase, but we in the meantime did it also with other photorespiratory mutants. It partially rescues the mutant, which indicates that indeed this bypass overcomes some of the limitations of photorespiration in a mutant background, but apparently does something in the wild type that is not beneficial. I don't show you the data. Why did? Maybe I did do? No. I've actually moved out the slides about the metabolomics and so on. It has to do with withdrawal of too much carbon into organic acids. So we shift too much in the direction of oxaloacetate, malate, and aspartate, and there's not enough carbon shuttled back into the Kelvin-Benson cycle, and that is why they don't grow as well. Okay, so then we thought this didn't work well in peroxisomes. Now let's just put it into the chloroplasts again on one construct. Here we have our, and in addition, we need the glycolate dehydrogenase because the chloroplasts cannot convert glycolate to glyoxylate. So we introduced a glycolate dehydrogenase so that we have now 6 enzymes on one construct and transformed into Arabidopsis. And we do get indeed some lines that grow better than the wild type. May not be totally obvious from the from the pictures, but here we have the growth curves and the rosette area. In gray, this is the wild type. The dark blue and light blue here are 2 constructs that grow approximately like the wild type, and we have 3 that are growing faster than the wild type and are achieving a larger final rosette size. This pathway also suppresses the phenotype of the transporter that exports glycolate from the chloroplast. So we do get better growth with the BHAG. Then we measured the carbon compensation points, the Amax, the respiration in the light, the respiration in the dark. There's no big difference in the carbon compensation point. It's not statistically different, but what we see is a lower Amax, which is a little bit unexpected because actually these are all the plants that I think 2 guys or 3 guys here are growing better than the wild type. So you would think a plant that grows faster than the wild type should have a higher Amax, but it doesn't. Well, what is Amax? Amax is measured under saturating light and saturating CO2 concentrations. So this is the total capacity of Rubisco. So what this number indicates is that these guys here have a lower capacity of Rubisco CO2 assimilation as compared to the wild type. And our assumption now is that these guys simply make less Rubisco. I'll come back to this thing later in the talk. In the meantime, a Chinese group has skipped this, what's called, not skipped us, scooped us, and put, and I don't know how they did this, because we published it in 2023, right? And it was like, nobody knew about this pathway before. The patent was also like filed later. So it's totally unclear to me how you can do all these constructs, all the field trials like within that time. So it's anyway, but the interesting thing is that they find that the effective tiller number is higher in the BHAC lines. So that's what they call BHAC, and it's a different version here. This has a different aminotransferase thing here. Here's the biomass production per plant, it's higher, but it's like not so interesting. What I found more interesting here is the grain yield per plant in grams at low nitrogen and high nitrogen. And what you see is like in the wild type, you get a massive decrease in the grain number per plant under low nitrogen. And this difference is much lower in the plants that express the BHAC pathway. And I think this potentially relates to the finding that I showed you here. So they have potentially less investment into Rubisco, which is the major nitrogen sink in the leaf. And so this, they are a little bit more nitrogen use efficient. Okay, so now coming from the BHAC to something that is, I think, even better. And I'm waiting for the Chinese paper coming out on this very soon, I guess. And I don't mean to be mean here, but I still think it's somewhat strange. So can we convert directly phosphoglycolate to phosphoglycerate while incorporating CO2? We possibly can, because the enzymes that do that have been developed by Toby and Aaron. Toby Arab in Marburg at the MPI, and Aaron at the time at the Max Planck in Golm. So they developed this new to nature carboxylation pathway in which glycolate is first activated to glucolyl-CoA at the expense of ATP. Then we have this new enzyme, the glucolyl-CoA carboxylase, that carboxylates glucolyl-CoA and makes tacronyl-CoA. And then tacronyl-CoA is reduced to tacronate semialdehyde by another new enzyme. And then we go to glycerate and can go back into the calvin-benson cycle. So if this indeed works, we are implementing an additional supplementary carbon assimilation pathway that is based on a production of phosphoglycolate, which is pretty cool. And we put that this time directly into the chloroplast, of course. And this is how it looks like. We put it onto two different constructs because the construct would have become too large to put it into a single tDNA. At least it didn't work in Arabidopsis for whatever reasons. And again, we get larger plants. This is, you can see it in the rosette size, but it's maybe more apparent in the quantification. So here is the wild type growth curve over time and the final rosette diameter. So we reach approximately 15 centimeters area in the rosette in the wild type and 30 centimeters in three independent transformed lines. And this is the final biomass of the rosette in dry weight. 20 milligrams in Columbia 0 and around 50 to 60 in three independent transgenics. This is quite substantial. So a doubling or tripling of the biomass in Arabidopsis, I think, is quite amazing, and it's statistically significant. Again, we see a similar thing here. So here's the maximum assimilation rate in Columbia, and here is three independent transformers, and they all have lower Amax. The carbon compensation point is not much different, but we also see lower respiration in the light and a slightly lower dark respiration rate. So again, this carbon to nitrogen thing seems to happen. And this is really important. Coming back to a slide that Loic has showed in the beginning, where you showed the increase in yield over time. So the yield increase over time kept up with population growth. So we tripled yields since the 1960s. But this came at the expense of a 7.3 increase in nitrogen fertilizer consumption. So we can keep up with population growth, but the amount of seeds, whatever yield that we produce, is not only genetic gain, it's a lot more fertilizer that goes in here. Or the genetic gain is only realized in the presence of a lot more fertilizer. And so if we want to really overcome some of the problems that we are facing, we not only need to increase yield, but we also need to increase yield with lower input. And of course, our plants can contribute to this because they have, otherwise I wouldn't show it. Well, we also looked at the carbon to nitrogen ratio and the delta 13C in the rosettes of these plants. The carbon to nitrogen ratio in the rosette is not significantly different. So we need to actually look at protein amounts and also look at nitrogen assimilation rates and things like that. So it needs deeper analysis to go into this. The delta 13C is slightly higher in the rosettes. And in some lines, or at least in 2 out of the 3 lines, it's also higher in the seeds, which the seed carbon integrates over lifetime. So it's a good estimate of lifetime water use efficiency. And the higher delta 13C in the seeds indicates that they actually might have also a higher water use efficiency. Again, this needs more analysis. It needs also field testing, of course, because it's all greenhouse data. But it looks very promising. The carbon to nitrogen ratio in the seeds is also more or less unchanged. So seed numbers and seed yield and so on, we still need to look at. Okay, so what's next? And then I will be at the end of the talk. Of course, we need to test this in the field, and we're trying to do this with sunflowers. Why not tobacco or potato or something? Because sunflower is the symbol of the Green Party in Germany. And we felt it would be a good idea to make the symbol of sustainability and ecology even more green by making it a bit more efficient. And the other factor is that the collaborator in this project is Corteva, and they wanted us to work on sunflowers. Otherwise, I would have used any other plant than sunflower, because sunflower is a pain to work with. Anyway, if it works and we get the transgenics, and I think we have some, they will go this year into, in 2025, yeah. They will go to the protected site in Switzerland. These are collaborators in the project, and they can do the field trials. So I think we can turn photorespiration into a carbon positive process, and through this overcome some of the inefficiencies. I should thank the people who did the work. These are the people from my lab, with the exception of Benjamin, also at Düsseldorf. And these are the collaborators in other labs and in other universities. And I should particularly point out Arn's group and Toby's group, who are really the enzyme wizards. They have engineered these enzymes, have come up with the ideas for this. And without this input or without these collaborations, I would not be able to show you anything here other than C3, C4 intermediates. Okay, thank you.
Yeah, thanks a lot.